Can dealing with protein workflows be easier, less frustrating, and produce better yields? Well, yes, but often it comes down to the equipment and the kits used by African scientists. Whether you are a researcher focused on expression, purification, and characterization of proteins, or you're looking at more commercial endeavors around therapeutic manufacture as well as diagnostic manufacture, Separations has a plethora of solutions available to support you. In this interview with Alicia Moodley, we're going to go through those world-class solutions, and who knows, you might just uncover a piece of technology that can make a massive difference in your lab.
Hi, today we're going to have a discussion with Holly regarding protein production and purification, essentially looking at everything through the isolation, the characterization, and seeing what services Separations can offer to support this workflow and help enable South African and African researchers in the space. Holly, great to have you on. Could you take us through a bit of this discussion?
Thanks, Jeff. Happy to be here. Thank you for having me. So welcome. We're doing a session on the protein biochemistry workflow. Within this workflow, we can break this up into many different techniques and approaches that researchers can use to be able to answer their research question. This is just going to be to build a better understanding of the protein biochemistry workflow, the different products and solutions we can offer around these different techniques. So let's jump into it. So we're going to cover some of our key suppliers. At Separations, we work with an array of different suppliers to bring support and their technology into the country. And we're going to cover some of the key suppliers specific to protein biochemistry. As I've mentioned, we can break some of the protein biochemistry workflow into different techniques. And we're going to take a very step-by-step approach to be able to show you the different solutions we can offer by each protein technique. So if we take an overview of our key suppliers, on our left, we have a range of suppliers that we're able to offer a range of consumables for the protein biochemistry workflow. This is an array from reagents to purification columns to filter papers. On our right, we have a range of suppliers that we offer equipment for the protein biochemistry workflow, and this can range from plate readers to liquid handlers to mass specs. Within the protein biochemistry workflow, we have broken this into 6 different techniques that we typically see. The first is production and expression, and then we follow this by extraction and purification. After extraction and purification, we can move into sample prep and sample cleanup. And then we move into some downstream processes, which is assays and proteomics, followed by detection and visualization. So we're just going to do this to provide a much clearer understanding on the different approaches researchers can take and where our solutions can fit in. We're also going to go through very step-by-step and then run through the different solutions we're able to offer.
Awesome. And Holly, I know you do this at the research scale with a lot of universities and biotech companies. But I understand that a lot of this is also for production. So if someone's looking to go to a diagnostic or therapeutic product, they can also do this under like the GMP guidelines, so good manufacturing practices. They can implement these same principles from lab all the way through tech transfer to production and manufacturing.
Yeah, absolutely, Jeff. We have a wide range of solutions that we can offer from all the way from lab scale all the way to production. Even into GMP compliant facilities. And as we're going through the different techniques, I'll show you a few different products that we that fit into that application, specifically in production, and how we're able to offer and what solutions we're able to offer there.
Awesome.
So if we jump into it, we're going to look at protein production and expression. And what do we mean when we say this? Well, it's the process of being able to generate, synthesize, or modify our protein of interest in living organisms. So why do we do this and how do we do this? So if we look at some of the applications of recombinant proteins, there are an array of reasons we want to be able to look at recombinant proteins, and some of these popular reasons are for the development of therapeutics, to look into the production of vaccine. We also do structural studies and enzymatic studies of enzymes, so we can see how we can use this to our benefits. So how do we begin this process of production and expression of our protein of interest? Well, it generally starts with the target gene, a protein of interest in DNA sequence form or in an oligo encoding for our protein of interest. We then take this and ligate this into an expression vector. An expression vector, we have those different ranges of expression vectors dependent on the type of approach you want to take. This can range specifically based on your host cell, and we have different promoters in expression vectors based on host cell. It can also be dependent on different features that you're looking for. So for instance, if you're interested in adding an epitope tag at the C terminus or the N terminus of your protein for later purification, that is something you will determine with your expression vector. There are also other specific modifications and expression vectors that you can build into your experimental design for downstream analysis that you're able to do. So once we ligate our oligo or our protein of interest into the expression vector, we then transfect it into a host cell, where it goes through the central dogma process of being translated from DNA into our functional protein. And then once we get our functional protein, we're able to do downstream applications with this. So if we look at an overview of the products that we're able to offer for protein production and expression, when it comes to consumables, we're able to offer a wide range of the transfection reagents and the expression vector systems. When it comes to transfection reagents, it's really important to be able to determine the cytotoxicity and the biodegradability of your transfection reagent. You want reagents that are going to be as less cytotoxic to your cells as possible to be able to have a much higher transfection efficiency. A higher transfection efficiency means that you have more protein being produced in your cell. When it comes to expression systems and expression vectors, we offer a wide range based on your host cell, based on the different types of modifications that you're looking for, and it's really going to be dependent on your application, and we can work closer with you to be able to help you determine which expression system is the best for you.
Awesome. And then on that point, Holly, I mean, I know that we often get involved with commercial cell lines as well, where I know that we have some suppliers that support not only offering a commercial cell line, but potentially also doing some modifications to an existing cell line to make it more commercial, and then go through that process of qualifying that cell line for commercialization. I believe that's also a service that is possible in-house.
Yes, that is definitely something that we're able to offer. And when it comes to specific workflows or specific experimental designs where you need specific knockout cell lines, that is something and an approach that we can take if you're looking at silencing RNA, all these different types of approaches. That's something very more catered or precision type of approaches. That is something we can offer, and we can work with you to be able to offer those here.
Awesome.
Another thing is, if you aren't working with a host cell and you're not the kind of facility that has a cell culture facility in place, then we can offer something called the human cell-free protein system. And if you're interested in your protein expression, this allows you to do it in an in vitro manner. The limitation with this is that you don't have the natural resources of being able to go through the process of things like protein folding that you naturally get from a host cell. But we do offer protein folding kits that help to ease this limitation. If you are the type of facility that has host cells, we offer a wide range of bioreactors for the production or the growth of your host cell. We offer everything, and this is where we offer everything from lab scale all the way up to production scale. And we're able to help you determine which is the best based on your application.
No, awesome. And just to add on that, I mean, essentially everything from stainless steel bioreactors and fermenters through to single use, completely validated traceable systems that just lower the requirement in terms of cleaning validation. So everything from pretty much research, tech transfer to commercial production for what we would see as a therapeutic product. Great.
Perfect. Thanks, Joe. So if we move on to the next protein technique, we'll start looking at extraction and purification. So once we have expressed our protein of interest in the cell, our host cell, we look at different methods to be able to extract and purify that out of our host cell. So what does this look like? So if after the process of translating into our protein, there's the general stage of cell lysis, where we use a lysis buffer to disrupt the membrane and be able to extract our protein out of the cell. To then further do purification, depending on your downstream application, we generally run your lysate through another column to be able to further purify and to try obtain your protein of interest. This can be done in many different approaches, and it's really dependent on the type of analysis you want to do downstream. So the different types of approaches that you can use is size exclusion, where your column, you separate your protein of interest from the rest of your cellular proteins through molecular weight. You can use ion exchange to be able to separate based on charge. And then you can also use affinity binding, where you have a chelating agent, such as a ligand or an antibody that is specific for your protein of interest. When it comes to a ligand, it has an affinity for your protein of interest. This can be, for instance, like we spoke about the epitope tags. You can have a ligand that has a specific binding affinity for your epitope tag. For instance, histidine, if you have a column that is a nickel column, it has a high affinity for histidine, or biotin for streptavidin, and we can use the strong binding affinity as an approach to be able to pull our protein of interest down. So what you'll do is with an epitope tag, for instance, you'll have it at the C terminus or the N terminus bound to your protein, and you use that almost as a label to be able to pull out your protein of interest.
Awesome, excellent. I'll add something here, because this is often where I see like between that research transition to larger manufacturing. We typically, because size exclusion and affinity can be a bit expensive, we often develop methods around the use of ion exchange or hydrophobic interaction initially, and then kind of look to size exclusion towards the end. So that's quite a nice dynamic that I see between dealing with research scale and the commercialization, is to actually consider how expensive those sorbents are, or those resins are at scale, and how many cycles you can get through it. So especially if you're doing something repetitively and you want to purify consistently for commercial outcome, that cost of not only the resin but also the solvents used, it really gets into a lot of optimization. So it's really nice that you highlight the different affinities and the different selections, because it's so easy to just want to use size exclusion or affinity and make it easy, but sometimes you've got to consider that commercialization step and those kind of considerations. Cool.
Yeah, absolutely. And when it comes to things like that, time is also something to really consider. And sometimes going to a faster approach, and we'll touch on some of the products that we offer, but using a system that you're able to automate this process. So when it comes to the purification steps, you also have antibodies, which is also very specific. That can be an antibody that is raised against your specific protein of interest that you can use to be able to pull it out of solution. So the typical process is that you have your lysate, you run it through your column. Your column has an affinity for your protein of interest based on the specific property that you chose up with your column, and it will bind to your protein of interest, and you'll be able to elute out all the other cellular proteins or proteins that you're not interested in, and then you can elute out your protein of interest. So if we touch on the products that we're able to offer, from an extraction point of view, we're able to offer lysis buffer, where you can do just the basic lysis of your cell, disruption of your cell to be able to get out your crude protein. And then we can move into the more specific purification. And this can be in the form of a resin. It can be in the form of filter-based membranes or filter-based columns, or in the form of magnetic beads. So when we're talking about upscaling and moving into production, things like magnetic beads, this can be fully automated. This entire process can be fully automated. So you have your magnetic bead bound to your specific chelating agent. So let's say nickel, for instance, and you then have it in your sample and it binds to your histidine tag. You then use a magnetic bead to be able to pull your sample, your protein of interest, attach that magnetic bead out of solution. That process can be fully automated. That process can be automated in line with things like a liquid handler, for instance, and then upscaling that for high throughput of that for high throughput samples becomes something that's a lot easier, saving time. So those are one of the benefits of using magnetic beads. With resin, we offer with resin columns, we offer a wide range. It can be pre-packed columns. We offer it in bulk. So it really is dependent on your scalability and your downstream applications and what you're trying to do. We also offer so continuous flow, or if you're looking at larger scale or higher throughput, we also offer things like reactor systems for purification processes. And then we also have the filter-based systems where you can use size exclusion, and this can be in the form of centrifugal tubes or filter plates. So once again, it is quite dependent on what your downstream applications is and what your purification, what type of purification method you're wanting to use, what type of chelating agent you're wanting to use. But once again, this is an experimental design, and there's so many different ways to be able to create a great solution. So if you reach out to us, then we can assist step by step throughout this entire process.
Yeah, and on that point, I'd like to highlight, I mean, I see the use of centrifugal devices. Often what we see is the commercialization that moves to the use of like tangential flow filtration, where essentially it's not like direct filtration where you block a membrane, you're constantly in flux and you're circulating solution, and you can use it for clarification as well as diafiltration, concentration. But it's that method of being able to, at large volumes, you can't just keep centrifuging. You've got to start using newer technologies, and that's often where we have this consultative role with clients, and we review like, yes, maybe centrifugation works at bench scale, but if you're going to be producing liters, thousands of liters of the product, you're going to have to look at technologies like TFF, tangential flow filtration, and also you might use one or two of these actor type systems where you might need to use ion exchange, and you need to do like pKa shifts, which means you do a lot of buffer exchanges, you've got to do some mixing, a lot of this needs to be maintained aseptically. So those are some of the solutions we try to always integrate when we talk about a comprehensive overview of protein extraction and purification.
Thanks, Jeff. So if we go into the next protein technique, we look at sample prep and cleanup. And this is one of, as Jeff mentioned, TFF, this is one of where it also plays a role. So this process is trying to improve the quantity or concentrate our protein samples or remove agents that affect our downstream processes. So if we look at this, this can be done. So after our purification process, we can run this through another column. This takes many different forms, and depending on your scalability, it can look as though it's a column. It can be a TFF system, capsules. It really is dependent. But for the sake, we're just going to look at an approach just based on the column and the concept on a column. So what happens is that you run your lysate through the column, and it's the column that has an affinity for your contaminants that you're looking for. So then your contaminant will then bind to the column, and then you can elute your protein of interest out. And our general contaminants are things like salts, lipids, small molecules, and detergents. This generally comes from the buffers that we're using, the lysis buffer, our eluting buffer, all of these different things, as well as cellular debris that also comes through. So if we look at some of the products that we're able to offer for sample prep and cleanup, when it comes to sample cleanup and concentration, there are many different things that we're able to offer, from centrifugal devices from Pall, and also this TFF systems from Cytiva and from Pall. When it comes to sample cleanup and sample prep for downstream applications, so specifically things like mass spectrometry, we look at things like HELIC, which is to be able to remove things like salts. We do magnetic beads bound to things like trypsin or chemotrypsin, where it's for the digestion or sample prep of your proteins to peptides for mass spec. We offer a wide range of columns from Phenomenex, SBE columns, columns for the cleanup of phospholipids, beta-glucuronidase, and different filtration systems for the cleanup of your samples for your downstream applications. If we look at some of our downstream applications, and this is where we get to after we've done some of our sample prep and cleanup, one of the applications is looking at assays. And assays is just a process of being able to analyze our substance or our protein of interest and we use it to be able to determine composition, presence, or just the general functionality of our protein. So once we receive our protein of interest, there are many different assays that we can use and different enzymatic assays and approaches that we can use, and that's dependent on what you're looking for, essentially. When it comes to assays, there are also different methods. There's using absorbance, luminescence, fluorescence. There are different types of kits based on these different methods, and all of them can be used to be able to determine different things about your protein. And some of the things that we look at are things like concentration, the interaction of our protein, the activity of our protein, and this helps us determine a lot of the functionality of our protein. So if we look at some of the assays that we're able to offer, a wide variety and different range, it really is going to be dependent on what it is that you're trying to look for, what it is you're trying to, what kind of questions you're trying to answer. But we do a wide range of cellular biology assays, caspase, apoptosis, cell viability, calcium, and we also do for if you're looking to do protein quantification, we have an assay that will help you be able to quantify your protein of interest. And we find that a lot of the time, and especially in drug discovery facilities or research labs that are doing a lot of assays, we start to move into an approach where you have a very high throughput of samples coming in. And one of the things that we're able to offer from Hamilton are liquid handlers, where we're able to automate the process of your assay development.
Yeah, awesome. I mean, I'll say that. I mean, we noticed a massive uptick in terms of South Africa, in terms of the use of liquid handlers, specifically even with the diagnostic sector for specifically the COVID testing. So it's actually great to see the presence of so many liquid handlers in Africa, and the adoption in terms of just getting precision and repeatability of results, and also just being able to work unattended for, through the evenings. So fantastic to see that, and hopefully we'll see more of these protein platforms going onto liquid handlers as well.
Yeah, and the great thing about the liquid handlers is that with Hamilton we have, and once again, we're able to offer so many different wide ranges. And with Hamilton, we have even people who are just interested in entry level, getting into entry level of automating, or getting into the start of automating their labs or samples. We have smaller systems that we're able to offer.
Awesome.
It's not just these there's not just these high-end systems. So the next technique that we're looking at, or downstream application that we're looking at, as proteomics, and this is quite popular. We're trying to after we've isolated our protein of interest, and it's the process of being able to study our protein further, study large-scale proteomes, and then to be able to understand their function. So if we look at this, it's our protein of interest, and it's really trying to get into the bare bones of the peptides, the amino acids, being able to understand the structure, tertiary structures, and get a better idea of the functionality, binding sites, all these different characteristics of our protein. And if we dive into it, there are many different systems that we're able to offer. So when it comes to equipment-based systems, we have EvoSep, which is really great, and especially designed for clinical facilities. So when it comes to mass spec, it's the front end of mass spectrometry. It's a standardized platform designed for the front end of mass specs, and we use this for high throughput sample prep for mass specs. Everything's essentially done on deck on the EvoSep, and peptides are effectively retained, and with very minimal to no cross-contamination. So just reproducibility in facilities like that prior to mass spec. And then when it comes to mass spec, we use this to be able to identify unknown compounds within a sample, and help us to be able to understand the structure and the chemical properties of proteins. When it comes to phenomics, we're able to offer columns for further peptide analysis, and these columns are titanium based and have no active sites in the column. And from Resin Biosciences, as mentioned, we do the HELIC as well as the trypsin-based beads, which then allow us to do the sample prep prior to mass spec.
Awesome, excellent. And I believe we've got quite a massive install base within Africa. I think arguably one of the biggest regarding the CyX equipment. I think close to 100 units in terms of under service and maintenance. So definitely a fantastic detector, fantastic technology.
Yeah, absolutely. Couldn't agree more. When it comes to our last technique, we're going to look at detection and visualization. So this is the process of actually being able to observe and characterize the structure, the quantity, or the interaction of our proteins. So once we look at our downstream applications, so be it assays or proteomics, one of the things that we want to do is actually be able to visualize and detect our protein of interest or these different downstream applications. So for instance, for assay-based work, if we're doing absorbance, how do we detect that? If we're doing fluorescence or luminescence, how do we end up detecting that? What platforms can we use to be able to detect our assays? And then when it comes to mass spectrometry or proteomics, for instance, what kind of analysis do we use to be able to detect our protein of interest? What does it mean? And what is it? And how do we look at the specific characteristics? And some of those characteristics can be things like size, the concentration, the composition, activity, and the function. And generally, with at the end of our detection and visualization spectrum, it can be in the form of a graph or graphical, like for instance this, and this gives us and provides us with so much valuable information that we can then determine about our protein of interest. So if we look at the different products that we're able to offer for detection and visualization, so on the front end, or just before we go into things like mass spec, if we're just looking, once we've purified our protein, for instance, and we want to just do a quick visualization of our protein based on molecular size, what we offer is a wide range of Western blotting, membranes, filter papers, Western blotting reagents, fluorescent proteins, just to be able to see if your protein is present. So the way that this works is that you run a Western blotting, SDS gel, and then you transfer your proteins onto a Western blot. This still keeps the activity of your protein, which allows you to then add a primary and a secondary antibody raised against your protein of interest, and then be able to detect and visually see that your protein is present, or being able to determine the molecular weight of your protein. Prior to that, so with assay-based approaches, we can look, we have detection platforms, plate reader platforms for our assay-based approaches. So molecular devices, where we're able to offer wide range, everything from absorbance, fluorescence, luminescence, TRF, fluorescence polarization. There's just a wide range that we're able to offer. And one of the best things about molecular devices is that we can offer GXP solutions. So if you're moving into platforms, if you're in pharmaceutical industry, if you're a GMP compliant or need to be GM compliant facility, we offer a wide range of solutions for this. When it comes to observing our protein of interest, something we can use is a fluorescence microscope. So for instance, when you're at the transfection phase, and you've transfected your cells and you want to see if you got a really good transfection efficiency, one of the ways that we can do that is with fluorescently labeled proteins. We take a sample of our cells and we stain it for fluorescent labeled antibodies, and we run that onto a microscope, and we can do an analysis of how many cells are fluorescently labeled and determine our fluorescence, our transfection efficiency using this. Also detection of a protein. We can also do interaction studies. So if you do confocal fluorescence microscopy, you can do co-localization studies where you can take one protein of interest and another protein of interest and have two differently fluorescent labeled, different labels on it, and then see if they co-localize into the same place in your cell. There are many different applications when it comes to fluorescence microscopy. then we can offer a wide range of visual and detection platforms from SciX, our mass spec, and for both Helena and SciX, we can do capillary electrophoresis. And and then from Cytiva, we also offer BioCore, which is a real-time label for interaction analysis to be able to actually measure the affinity or the kinetics of your analyte and your ligand binding.
So I'll jump in there. I mean, the BioCore products are very powerful in terms of biosensors when you're looking at association curves, dissociation, the kinetics, the binding, really just looking at the functional aspects, specifically around binding and dissociation. And the nice thing as well with the Leica products, I mean, I noticed that, in the separations, demo lab or, the working wet lab, there's actually quite a range of options from the microscopes placed there. So customers are always encouraged to come through a sample, have a look functionally, how it works, get a sense for it practically. And then that also helps sometimes with making a decision in terms of which platform is going to work the best for you.
Absolutely. And when it comes to Leica, they're highly modular. We're able to build these systems to meet different applications, different approaches. So we can build these systems to be more tailored to the kind of approach that you're working with. The different fluorescence, for instance, with fluorescence, you're working with different fluorophores. So whichever system that you're working with, whichever wavelengths that you're working with, we can tailor these systems to that. Very much tailored to your experimental design. Okay.
Awesome.
So that covers all the different protein techniques and an overview of all the different protein techniques that we can go. And I hope that gives everybody a little bit of a better understanding of the different products that we're able to offer from separations. Once again, we're always here to assist, and we're always here to be able to help pick and provide the best solutions tailored to your application and the type of work that you're trying to do, the type of experimental design that you're looking for, all the way from from the production and expression all the way down to detection and visualization. This entire workflow, we are able to offer a full workflow solution. So you just reach out to us at any point in time. Within separations, we have many different divisions, and they overlap quite a bit when it comes to the different products, but some of the different products fall within the different divisions. So please feel free to contact the division of interest if you find that there's a product that you're specifically interested in.
I'll add in there. I mean, essentially, a lot of these divisions work very closely together, but they all specialize. So, you could have like multiple team members within the chromatography division, and they specialize just in chromatography. And it's nice to have this overview of like different levels of expertise, but it's great to be able to kind of bring subject matters in on each project and highlight an aspect. So like whether it starts in the initial lab with, defining the cell line, characterizing it, getting it going, taking that to commercial levels in terms of, bioprocessing, biomanufacturing, pharmaceutical aspects. And the nice thing is, even like we didn't talk about it, but there's full and finished solutions. There's all these QC, quality control considerations. And then we've got teams like robotics that's really focused on automation, which is how to improve throughputs. And then all these different subject matter experts. So, always trying to bring the most to create like raving fans in terms of dealing with the products. And it's just a really comprehensive presentation. So, Holly, I can only say thank you so much for so much detail, so much effort you put into to kind of highlight these different synergies of products and how they all come together. And we're looking forward to producing more content that highlights and showcase specifically general topics like protein biochemistry, but it's quite intricate. There's quite a bit of specialization that goes into it, and hopefully this presentation created a bit more awareness of the in-house capabilities and what we can support within separations.
Yeah, perfect. Thank you, Jeff. And if you don't know where to start, then just email info@separations.co.za, and we'll be there to help you.
Absolutely. Absolutely always enthusiastic to get started on a new project and help customers and clients and scientists ultimately in the South African and African region. And a lot of these distributors we've mentioned are exclusive to us, and we enjoy working on these workflows. Thank you again, Holly.
Thank you, Jeff.
Now, I really hope you enjoyed that interview, and if you are as passionate about science as we are for Africa, please check out our YouTube channel for additional content.